purpose of these episodes not at all for advice or medical suggestions. These are aimed to provide support to peer pharmacists in training in educational and intellectually stimulating ways. Again, these are not at all for medical advice or medical suggestions. Please see your local board and state-certified health professional. The views of this podcast represent those of my guests and I.
Well, it's not so much about the environment being there already, but you can complement to creating that environment for yourself.
I think that's key, yes.
Didn't have a map, just a pencil in my hand. Good at science, good at math, trying to understand. They said be an engineer, so I followed that advice. Found a spark in a chemistry class, didn't have to think twice. Wasn't all planned out, just a search for something new. Helping start a school for kids with a different point of view. I meant to live the words that he would never have to say. He showed me that the biggest walls
Able to thrive scientific intervention. I heard someone say you can almost somewhat bend the environment to your will or complement it, change.
And sometimes melt away, he said, go on and dream those big dreams. Paint a whole new shade of sky. Believe that anything is possible and give it a real try. 'Cause you're gonna that stubborn beating in your heart is what it's all about. So don't you give up. No, don't you give up now. It's easy doing incremental, adding piece by piece, but a transformative idea gives the world a new release. So I try to hire nice people who want to.
If you have a big effect on the environment by the way you treat people and the way you act as a role model or lack thereof.
That's true. It's very big reaction.
Make a change, build a team that works together to rearrange the page. From nanoparticles fighting the pandemic's awful sting to tissue engineering the new life that we can bring. It's more than just the accolades or prizes on a shelf. It's the environment you're building, bigger than yourself. Remember, go on and dream those big dreams. Paint a whole new shade of sky. Believe that anything is possible and give it a real try. 'Cause you're gonna find the roadblocks, you're gonna hear the doubt, but that stubborn beating in your heart is what it's all about.
In terms of you maintaining the view of the bigger picture in your career and in your life in general, how have you done that? When you face challenges or obstacles?
Don't you give up. No, don't you give up now. My wife says be home by 7, keep a balanced point of view. Never be gone for too long from the ones who count on you. It's the bigger picture calling through the static and the noise. It's the people you empower in their triumphs and their joy.
Welcome to The New Chemist. We're glad you're listening. Feel free to download this podcast on Apple Podcasts, Google Podcasts, and Spotify. Here on The New Chemist.
Didn't have a map, just a pencil in my hand. Good at science, good at math, trying to understand. They said be an engineer, so I
community, research, and COVID-19. We're happy you're tuning in. My guest today is Dr. Robert Langer. Thanks for joining me today. It is good to hear from you.
Followed that advice, found a spark in a chemistry class. Didn't have to think twice, wasn't all planned out, just a search for something new. Helping start a school for kids with a different point of view. I meant to live the words that he would never have to say. He showed me that the biggest walls can sometimes melt away. He said, go on and dream those big dreams, paint a whole new
Inventor and one of the 12 Institute professors at the Massachusetts Institute of Technology. He was formerly the Germershausen Professor of Chemical and Biomedical Engineering and
shade of sky. Believe that anything is possible, and give it a real try. Cause you're gonna find the roadblocks, you're gonna hear the doubt, but that stubborn beating in your heart is what it's all about. So don't you give up, no, don't you give up now.
of Biological Engineering at MIT. He is also a faculty member of the Harvard MIT Program in Health Sciences and Technology and the Canc Institute.
It's easy doing incremental, adding piece by piece, but a transformed idea gives the world a new release. So I try to hire nice people who want to make a change, build a team that works together to rearrange the page. From nanoparticles fighting a pandemic's awful sting, to tissue engineering the new life that we can bring.
Cancer research. In 2015, Dr. Robert Langer was awarded.
It's more than just the accolades or prizes on a shelf. It's the environment you're building, bigger than yourself. Remember, go on and dream those big dreams, paint a whole new shade of sky. Believe that anything is possible, and give it a real try. Cause you're gonna find the road.
Welcome, Dr. Robert Langer.
You're gonna hear the doubt, but the stubborn beating in your heart is what it's all about. So don't you give up, no, don't you give up now. My wife says be home by 7, keep a balanced point of view, never be gone for too long from the ones who count on you. It's the bigger picture calling through the static and
Okay, Dr. Langer, thank you so much for joining me today. It is good to have you here.
the noise, it's the people you empower in their triumphs and their joy.
Things that can hopefully have a big effect on the health and well-being of the world, and so, you know, in terms of teamwork.
Didn't have a map, just a pencil in my hand. Good at science, good at math, trying to understand. They said be an engineer, so I followed that advice. Found a spark in a chemistry class, didn't have to think twice. Wasn't all planned out, just a search for something new. Helping start a school for kids with a different point of view. I meant to live the words that he would never have to say. He showed me that the biggest walls
Encourage everybody to work together. You know, we have a lot of people, we have over 100 people in the lab, but I try to hire, I mean, this may sound almost silly, but I try to hire very nice.
and sometimes melt away. He said, go on and dream those big dreams, paint a whole 'Cause you're gonna find the roadblocks, you're gonna hear the doubt, but that stubborn beating in your heart is what it's all about. So don't you give up, no, don't you give up now. It's easy doing incremental, adding piece by piece, but a transformed idea gives the world a new release. So I try to hire nice people who want to,
but who also really want to make a difference in the world. And so that's what I've tried to do repeatedly. And they've been great. I mean, the people we've had in our lab have been outstanding.
Yeah.
Make a change, build a team that works together to rearrange the page. From nanoparticles fighting a pandemic's awful sting, to tissue engineering the new life that we can bring. It's more than just the accolades or prizes on a shelf. It's the environment you're building, bigger than yourself. Remember, go on and dream those big dreams. Paint a whole new shade of sky. Believe that anything is possible, and give it a real try. 'Cause you're gonna find the roadblocks, you're gonna hear the doubt, but that stubborn beating in your heart is what it's all about.
In terms of your success, I think by any standard of measurement, international, national, I think people can all agree that you have been successful as a pioneer in the field.
Don't you give up, no, don't you give up now. My wife says be home by 7, keep a balanced point of view, never be gone for too long from the ones who count on you. It's the bigger picture calling through the static and the noise. It's the people you empower in their triumphs and their joy.
Welcome to the New Chemists. We're glad you're listening. Feel free to download this podcast on Apple Podcasts, Google Podcasts, and Spotify. Here on the New Chemists, we discuss chemistry, which simply put is the science of change, as well as careers, community, research, and COVID-19. We're happy you're tuning in. My guest today is Dr. Robert Langer. Thanks for joining me today. It is good to hear from you. Just briefly, I'll inform my audience about you. Dr. Robert Samuel Langer Jr. is an American chemical engineer, scientist, entrepreneur, inventor, and one of the 12 institute professors at the Massachusetts Institute of Technology. He was formerly the Germershausen Professor of Chemical and Biomedical Engineering and maintains activity in the Department of Chemical Engineering and the Department of Biological Engineering at MIT. He is also a faculty member of the Harvard-MIT Program in Health Sciences and Technology and the Canc Institute for Integrative Cancer Research. In 2015, Dr. Robert Langer was awarded the Queen Elizabeth Prize for Engineering. He has numerous accolades and achievements. Please welcome Dr. Robert Langer. Okay, Dr. Langer, thank you so much for joining me today. It is good to have you here.
My pleasure.
Yes. So what has been some of the most beneficial advice you have received?
Well, I would say that, it's not so much advice as just a role model. Judah Folkman was my postdoctoral advisor, and he was the kind of person that believed anything was possible. I don't know if he used those words, but he lived that, and so I think that thinking that anything is possible, and I think recognizing when you think that way, you're going to often get criticism, there's going to be a lot of roadblocks, and then not giving up. And I think, he never said it quite that way, but that's that's kind of what I learned from him as a mentor, to dream big dreams and never give up.
Oh wow, that's very good. It's very good. Persistence is important. So, do you have any advice for those wanting to pursue the field you are currently working in?
Well, what I just said is part of the advice. I think it's important to do things that are transformative and not incremental, but recognizing that if you do that, you will run into roadblocks and you will fail sometimes. So, so the important thing is, as you just said, is persistence. Persistence is very important.
Yeah, that's true. Dr. Langer, I listened to several of your interviews, and one of the things that stands out to me is that your academic path, your academic journey was not easy. You encountered some challenges, and I think it's very commendable that you were able to persist and be successful amidst those challenges. So, in line with your major, why did you choose chemical engineering as a field to major?
Yeah, I would like to tell you that it was really well thought out, but I can't say that. Initially, when I was in high school, my dad and my guidance counselor said, well, you're good in science and math, so you should become an engineer. So I became an engineer, and then when I was at Cornell as an undergraduate, the course I liked the best and the one I was best at was chemistry. So I became a chemical engineer.
Okay. Oh wow. So
I'm glad I did. I think it's been a great profession.
Yeah, I would say so too. So in terms of your doctoral studies, is it along the same lines that you chose chemical engineering to do your doctoral studies in? Were those things the deciding factors, things that affected you in your undergraduate years?
Well, so I for a long time, I didn't have a very clear idea of what I wanted to do. As an undergraduate, like I say, I majored in chemical engineering for the reasons that I said. As a graduate student, in a way, it was just a continuation of that. I still was kind of searching for something I wanted to do. And when I was a graduate student, I got involved in a lot of other things too, like helping start a school for poor high school students and things like that. And developing new chemistry and math curriculum. And so it wasn't until my postdoctoral work that I really sort of found a research direction, and that was by being in the hospital. I worked in Children's Hospital as a postdoc, and I was really the only engineer there. And as I mentioned, I worked with Judah Folkman. He was a great role model, and I started to think about how I could solve medical problems with my chemical engineering background.
Great. Yeah, that's very good. So you mentioned how you started the school, and I remember hearing about the school in a previous interview. So my question to you is, why and how did you go about starting that school?
Well, actually, so my interest in teaching started when I was at Cornell because I was a teaching assistant, and I really loved that. And so when I came to Boston the next year and Cambridge to join MIT as a graduate student, I got involved in doing some tutoring activities in Roxbury and Cambridge. And so there were some people, Peter Haggerty, Neil Didrikson, Paula Didrikson, they were interested in starting this school for working class children, for kids. And they had heard about me, so they asked would I help on developing the math and science programs. And I thought it sounded like a very worthwhile thing, so I did.
Oh, that's very good. So how have you maintained vision and teamwork in your environment? How have you maintained those things in your work environment, in your lab? How do you maintain those things?
Well, I guess there are different things. So vision, I try to encourage everybody in the lab to think big and to dream big dreams and to do things that can hopefully have a big effect on the health and well-being of the world. And so, in terms of teamwork, and I encourage everybody to work together. We have a lot of people. We have over 100 people in the lab. But I try to hire, this may sound almost silly, but I try to hire very nice people who get along with each other, but who also really want to make a difference in the world. And so that's what I've tried to do repeatedly. And they've been great. I mean, the people we've had in our lab have been outstanding.
Yeah, that's very true. So, in terms of your success, I think by any standard of measurement, international, national, I think people can all agree that you have been successful as a pioneer in the field. So, what would you say has complemented the most to your success? Would it be mentoring? Would it be networking? Would it be your approach to studying or learning or teaching? What would you say has been the main driver or impetus in which you've been successful?
Well, I think to the extent that I have been successful, and that may be debatable, but to the extent that I have, I really think it's been the people. I've had wonderful, wonderful people working in the lab, and they've been great. I like to think that they feel like I've treated them well and that I've encouraged them to live their dreams. But really it's having just tremendous people. MIT's been a great place, and we've just had a tremendous number of very, very good students and postdocs in the lab. And, by any measures, I think 19 are now in the National Academy of Engineering. I think 16 in the National Academy of Medicine, and they've won all kinds of awards, started companies. I mean, they've been very, very successful. So to me, it's been them.
Yeah, sure. People are important, very important. So, given all your responsibilities and accomplishments, how do you strive to maintain a balanced life?
Well, my wife is very good at making sure I do. When my children were little, she told me that if I wasn't traveling and that I shouldn't, that I needed to be home by 7 every night to spend time with the kids. And when I traveled, I mean, of course, right now I'm not traveling, but when I did travel, certainly when the children were young, I mean, or even at home, I would never be gone for very long. I kind of learned that was important to them. So, just to give an example, 5 years in a row, I had to go to Israel for something, like a lecture, an award, or a degree, honorary degree. And all those 5 times in a row, 5 different years, I didn't use a hotel. I just flew over, I spent part of the day there, and then I flew right back. They have a 1 a.m. flight from Tel Aviv to New York, so I was able to do that. But anyhow, so I never would be gone for very long.
Okay, wow, yeah, family is a very important thing. So you would say your family is the basis on which you've been able to keep a balanced life.
Well, that, and my wife has been very good, Laura. She tells me what she thinks. So if I did something, if I was probably spending too much time doing other things, she would certainly let me know.
Okay, that's good. So, in terms of your impactful ideas, what would you say have been your most impactful and effective ideas to date?
Well, I think there's been 3 or 4 areas it's hard to, that have been important, I guess. One has been the work we did with Judah Folkman on isolating the first angiogenesis inhibitors, the first blood vessel inhibitors, and that's led, because of work that others had done after that, at companies and so forth, to many new drugs that would be useful in treating cancer and eye diseases. The second is probably the work on new materials and controlled drug delivery, which also, that includes nanotechnology, and that's also led to, quite a number of products by different groups again. And finally would be the idea of with Jay Vacanti of tissue engineering, where you could combine materials and cells and, make new tissues and organs, whether that be an organ on a chip or a new tissue in the body, like artificial skin.
Okay. So in terms of you finding and seeking the right environment for you to thrive scientifically and intellectually, how did you go about finding that? What was your guide, or what was your rubric or basis on which you found that environment? I take it was at MIT, but how did you find that?
Yeah, well, I mean, so there are different ways. I mean, I suppose the experience I had at Children's Hospital that was really key. And then MIT, I mean, that was, I guess, good luck. That they hired me, and things worked out there. Ultimately, though, you create your own environment. Really, when you run a lab, it's the environment you create for others. That ends up making a difference.
Yeah, it's interesting you say that, because I've heard other people say that as well. It's not so much about the environment being there already, but you can complement with creating that environment for yourself.
I think that's key. Yes, I agree. I agree with you.
Yeah, and you're able to thrive scientifically, intellectually. I heard someone say you can almost somewhat bend the environment to your will, or complement it, change it.
Well, you can create, if you have a big effect on the environment by the way you treat people and the way you act as a role model or lack thereof.
That's true. That's very good reaction. So, in terms of you maintaining the view of the bigger picture in your career and your life in general, how have you done that? When you face challenges or obstacles, how have you maintained a view of the bigger picture that kept you going, that kept you persistent? How did you do that?
Well, I guess that's just me. I'm a very stubborn person. If I believe in something, I just keep trying and trying. I don't give up easily. If I feel something can do some good, if I feel that we've made a finding that's important, I'll do the best I can.
Okay, yeah, that's very good. So, Dr. Langer, my career path, I will speak on my career path thus far. I started off in engineering at Georgia Tech, and some things happened. I transitioned to another institute, and I switched my major to biochemistry. So I changed path because also my interest changed as well. So my question to you is, for those in the field of engineering, what would be some of your advice, or what would be the key things that you did that complemented to you being able to transition to eventually a doctoral program in engineering and eventually to serve as a pioneer in the field? What were some of the key things that you did in your time as an undergraduate and as a graduate student?
Yeah, well, I think as an undergraduate, I feel you really want to learn the fundamentals. You want to learn the fundamentals, and you want to really do well in class. That should be your highest priority. I wouldn't do research or other things until I knew I could do well in class, so to speak. As a graduate student, I think then, you also want to do the same thing, continue learning the fundamentals well, but then you really want to pick a research project and a research advisor, if you're getting a PhD or even a master's degree, who's really a good mentor. And what does that mean? Hopefully that they treat you well and that they put you in an environment or have you in an environment where you can learn from them and from others.
Okay, yeah, that's good. So I think most of us can agree that this time has been somewhat challenging for a lot of people due to the pandemic. A lot of people have encountered obstacles or challenges. So what has kept you optimistic during this time, during this pandemic?
Well, two things. One of the things I've been doing is I've been spending a lot of time trying to fight this pandemic. One of the companies I helped start is Moderna. Moderna has been one of the leading companies in terms of creating the COVID vaccine. So they're now in what's called phase 3 trials for doing it. And actually, almost every one of the companies that's doing an RNA vaccine, and those are the leading ones in terms of moving forward in the clinic, uses nanoparticles, which is an area to deliver the RNA. And, so that's an area that we've done a lot on. And we're also doing work in the lab on vaccines, with masks, creating new masks, creating new ventilators, creating split ventilators, working on nasal sprays that might be able to combat it, working on new blood tests. So we've been very active in terms of really trying to hopefully make a difference in treating COVID. In addition to, even though research slowed down on other things because of the rules at the universities, I think that we continue that research too, and we're working a lot with the Gates Foundation on things that can help the developing world, and just other things that I hope will be important to the world at some point. And again, I believe in all these things, so we're not going to stop, and so, but all that is what we've tried to do.
So I have two more questions. In terms of your work with the COVID-19 pandemic, in terms of all the things you're designing and engineering, have you seen the impact, or is it still in the works? Have you started to see impact from your work thus far?
I'd say the answer to that is we've seen some impact. I mean, so what can I say? The fact that, some of the things that we did actually now over 45 years ago, creating, small particles that could deliver nucleic acids. Like I say, that's being used by Moderna, Pfizer, many companies. Again, lots of other people contributed too, but we did that early. We also developed other kinds of nanoparticles. And again, a lot of it's being done by our students in some of these places. So I think that the vaccines, they haven't made an impact yet, but other than the people that have been treated by it, that seems to me the nation's biggest hope, the world's biggest hope, is the vaccines. Other things we've done have had an impact. We've helped make new masks, and several million of those have already been used by people, and I think they're better masks. And some of the others are in the works, but are in clinical trials, and they've moved very fast. So I think that we've been able to make some impact on patients already, and hopefully more.
Okay. So, Dr. Langer, you mentioned, just going back, you mentioned how you said you're somewhat stubborn and you're consistent. Is it something that was taught to you in your upbringing, or is it something you developed over time in academic settings?
I think it's something that I just was born that way. I think I've always been stubborn. I'm sure my parents thought that.
Okay. Yeah, well, Dr. Langer, it was so good to have you on today. I really appreciate you taking this time to chat with me and for us to discuss your career path and how you're currently making a difference, how you made a substantial difference in chemical engineering and biomolecular engineering fields. So thank you again for joining me today.
Thank you. It's a pleasure, and good luck.
Thanks for listening. We're glad you were able to tune into this podcast. Once again, this is The New Chemist, where we discuss chemistry, which simply put is the science of change, as well as the other sciences, careers, community, research, and COVID-19. Thanks again for listening. Note, the views on this podcast represent those of my guest and I.
Una entrevista con uno de los más grandes líderes en ingeniería química, medicina y ciencia, el Dr. Robert S. Langer, SCD, profesor del Instituto Mítico fundador de Moderna. David Ferguson. El podcast del nuevo químico, una entrevista con David Joshua Ferguson, AMRSB-AMRSC, y el Dr. Robert Langer, profesor del Instituto Mítico fundador de Moderna. David Joshua Ferguson. Estamos felices de que nos esté sintonizando. Mi invitado de hoy es el Dr. Robert Langer. Gracias por acompañarme hoy. Y es bueno saber de ti. Solo brevemente, llamaré a mi audiencia sobre ti. El Dr. Robert Samuel Langer Jr. es un ingeniero químico, científico, empresario e inventor estadounidense, y mientras es profesor del Instituto Mundial en el Instituto de Tecnología de Massachusetts, es David Joshua Ferguson. Un profesor de Ingeniería Química y Biomédica se mantiene activo en el Departamento de Ingeniería Química y el Departamento de Ingeniería Biológica del MIT. También es miembro de la facultad del programa de Harvard y MIT en Ciencias de la Salud y Tecnología y el Instituto Koch para la Investigación Integrativa del Cáncer. David Josué Ferguson. En 2015, el Dr. Robert Langer recibió el Premio Queen Elizabeth de Ingeniería. Tiene numerosos reconocimientos y logros. Biografía abreviada. Robert S. Langer es uno de los 9 profesores del Instituto del MIT. Ser profesor del instituto es el honor más alto que se le puede otorgar a un miembro de la facultad. El Dr. Langer ha escrito más de 1570 artículos. También tiene más de 1400 patentes emitidas y pendientes en todo el mundo. Las patentes del Dr. Langer han sido licenciadas o sublicenciadas a más de 400 compañías farmacéuticas, químicas, biotecnológicas y de dispositivos médicos. Es el ingeniero más citado de la historia, índice H309 con más de 386.000 citas según Google Scholar. Se desempeñó como miembro de la Junta de Ciencia de la Administración de Drogas y Alimentos de los Estados Unidos, la junta asesora más alta de la FDA, de 1995 a 2002 y como presidente de 1999 a 2002. El Dr. Langer ha recibido más de 220 premios importantes. Es una de las 3 personas vivas que han recibido la Medalla Nacional de Ciencias de los Estados Unidos, 2006, y la Medalla Nacional de Tecnología e Innovación de los Estados Unidos, 2011. También recibió el Premio Internacional de la Fundación Gairdner de 1996, el Premio Charles Stark Draper de 2002, considerado el equivalente al Premio Nobel de Ingeniería, el Premio Millennium de 2008, el premio de tecnología más grande del mundo, la Medalla Priestley de 2012, el máximo galardón de la American Chemical Society, el Premio Wolf de Química de 2013, el Premio Breakthrough de Ciencias de la Vida de 2014 y el Premio Kyoto de 2014. En 2015, el Dr. Langer recibió el Premio Queen Elizabeth de Ingeniería. Entre muchos otros premios que ha recibido Langer se encuentran el Premio Dixon de Ciencias, 2002, el Premio Inc. de Tecnología, Economía y Empleo, 2003, el Premio Harvey 2003, el Premio John Fritz, 2003. Otorgado anteriormente a inventores como Thomas Edison y Orville Bright, el Premio General Motors Kettering para la investigación del cáncer, 2004, el Premio Dan David en Ciencia de los Materiales, 2005, el Premio del Centro Médico de Albany en Medicina e Investigación Biomédica, 2005, el premio más grande en el AS para la investigación médica, ingreso en el Salón Nacional de la Fama de Inventores, 2006, Premio Max Planck de Investigación, 2008. Premio Príncipe de Asturias de Investigación Científica y Técnica, 2008, Premio Fundación Warren Alpert, 2011, Premio Internacional Terumo, 2012, Medalla Benjamin Franklin en Ciencias de la Vida, 2016, Premio Caviller en Nanociencia y Nanomedicina, 2017, Premio Dreyfus en Ciencias Químicas, 2019, Medalla de la Ciencia, Máximo Honor de Portugal, 2020, el Premio Maurice Marie Janot, 2020, el Premio Fundación BBVA Fronteras del Conocimiento en Biología y Biomedicina, 2021, y el Premio Balzan de Biomateriales para Nanomedicina e Ingeniería de Tejidos, 2022. En 1998, recibió el Premio Lemelson MIT, el premio de invención más grande del mundo por ser uno de los inventores más prolíficos de la historia en medicina. En 1989, el Dr. Langer fue elegido miembro de la Academia Nacional de Medicina, en 1992 fue elegido miembro de la Academia Nacional de Ingeniería y de la Academia Nacional de Ciencias, y en 2012 fue elegido miembro de la Academia Nacional de Inventores. La revista Forbes, 1999, y BioWorld 1990, han nombrado al Dr. Langer como uno de los 25 individuos más importantes en biotecnología en el mundo. La revista Discover, 2002 lo nombró como una de las 20 personas más importantes en esta área. La revista Forbes, 2002, seleccionó al Dr. Langer como uno de los 15 innovadores a nivel mundial que reinventarán nuestro futuro. La revista Time y CNN, 2001, nombraron al Dr. Langer como una de las 100 personas más importantes de Estados Unidos y una de las 18 personas más destacadas en ciencia o medicina en Estados Unidos, America's Best. Parade Magazine, 2004, seleccionó al Dr. Langer como uno de los 6 héroes cuya investigación puede salvarle la vida. El Dr. Langer ha recibido 40 doctorados honorarios. Incluyen títulos de la Universidad de Harvard, la Escuela de Medicina Mt. Sinai, la Universidad de Yale, la Universidad de Columbia, la Escuela de Graduados Gerstner del Centro de Cáncer Memorial Sloan Kettering, la Universidad de Maryland, la Universidad de Western Ontario Canadá, Suiza, el Technion, Israel, la Universidad Hebrea de Jerusalén, Israel, la Université Catholique de Louvain, Bélgica, el Instituto Politécnico Rensselaer, la Universidad de Willamette, la Universidad de Liverpool, Inglaterra, el Bates College, la Universidad de Nottingham, Inglaterra, Albany Medical College, Pennsylvania State University, Northwestern University, Uppsala University, Suecia, Teléfono Aviv University, Israel, Boston University, Ben Gurion University, Israel, University of Laval Canadá, Carnegie Mellon University, Drexel University, Universidad de Andang, Corea del Sur, Universidad de Nueva Gales del Sur, Australia, Instituto Karolinska, Suecia, Universidad de Ciencia y Tecnología de Hong Kong, Instituto Nacional de Astrofísica, Óptica y Electrónica, México, Universidad de Limerick, Irlanda, la Universidad de Illinois, la Universidad Estatal de Ohio, la Facultad de Ingeniería Olin, la Universidad Alfred, la Universidad de Ciencia y Tecnología de Macao, la Universidad de Santiago de Compostela, la Universidad de Maastricht y la Universidad de California, Medalla de San Francisco. Recibió su licenciatura de la Universidad de Cornell en 1970 y su SCD del Instituto Tecnológico de Massachusetts en 1974, ambos en Ingeniería Química. Bien, doctora Langer, muchas gracias por acompañarme hoy. Es bueno tenerte aquí. Doctor Roberto Langer. Mi placer. David Josué Ferguson. Sí, entonces, ¿cuál ha sido uno de los consejos más beneficiosos que ha recibido? Doctor Roberto Langer. Bueno, diría que, ya sabes, no es tanto un consejo, es solo un modelo a seguir, ya sabes, Judah Folkman fue mi asesor postdoctoral y era el tipo de persona que creía que todo era posible. No sé si usó esas palabras, pero él vivió eso, ya sabes, y entonces creo que eso de pensar que todo es posible, y creo que reconociendo cuando piensas de esa manera, a menudo vas a recibir críticas. Va a haber muchos obstáculos y luego no darse por vencido. Y creo, ya sabes, quiero decir, él nunca lo dijo de esa manera, pero eso es algo de lo que aprendí de él como mentor, a soñar grandes sueños y nunca rendirme. David Ferguson. Oh, eso es muy bueno, muy bueno. La persistencia es importante. Entonces, ¿tienes algún consejo para aquellos que quieran dedicarse al campo en el que estás trabajando actualmente? Dr. Roberto Langer. Bueno, lo que acabo de decir, lo vi como parte del consejo, ya sabes, creo que es importante hacer cosas que sean transformadoras y no incrementales, pero reconociendo que si lo haces, hazlo, te encontrarás con obstáculos y fallar a veces. Dr. Roberto Langer. Entonces, ya sabes, lo importante es, como acabas de decir, la persistencia. La persistencia es muy importante. David Ferguson. Si eso es verdad, ¿sabe, Dr. Langer? Escuché varias de sus entrevistas, y una de las cosas que me llama la atención es que su camino académico, la formación académica no fue fácil. Te encontraste con algunos desafíos, y creo que es muy encomiable que haya podido persistir y tener éxito en medio de esos desafíos. David Ferguson. Entonces, de acuerdo con su especialización, ¿por qué eligió la ingeniería química como un campo para especializarse? Dr. Roberto Langer. Me gustaría decirte que estuvo muy bien pensado, pero no puedo decir eso, ya sabes, al principio, cuando estaba en la escuela secundaria, mi padre y mi consejero me dijeron, bueno, ya sabes, eres bueno en ciencias y matemáticas, así que deberías convertirte en ingeniero. Dr. Roberto Langer. Así que me hice ingeniero, y ya sabes, y luego, cuando estaba en Cornell como estudiante universitario, el curso que más me gustaba y el que mejor era el de química, así que me hice ingeniero químico. Dr. Roberto Langer. Me alegro de haberlo hecho, creo que ha sido una gran profesión. David Ferguson. Oh sí, yo también lo diría. Entonces, en términos de sus estudios de doctorado, es en la misma línea que eligió la ingeniería química para ver un estudio de doctorado. Son esas cosas, los factores decisivos, las cosas que te afectaron en tus años de estudiante. Dr. Roberto Langer. Bueno, ya sabes, durante mucho tiempo, no tenía una idea muy clara de lo que quería hacer como estudiante universitario. Como digo, me especialicé en ingeniería química por las razones que dije como estudiante de posgrado. Dr. Roberto Langer. Quiero decir, en cierto modo fue solo una continuación de eso. Ojalá todavía estuviera buscando algo que quisiera hacer, y cuando era estudiante de posgrado, también me involucré en muchas otras cosas, como ayudar a iniciar una escuela para estudiantes pobres de secundaria y cosas así. Dr. Roberto Langer. Desarrollo de nuevos planes de estudios de química y matemáticas. Así que no fue hasta mi trabajo postdoctoral que realmente encontré una dirección de investigación, y eso fue estando en el hospital. Trabajé en el Children's Hospital como postdoctorado y realmente era el único ingeniero allí. Dr. Roberto Langer. Y como mencioné a Judah Folkman, fue un gran modelo a seguir, y comencé a pensar en cómo podría resolver problemas médicos con mi formación en ingeniería química. David Ferguson. Sí, es muy bueno. Mencionaste cómo empezaste la escuela. Recuerdo haber oído hablar de la escuela en una entrevista anterior. Entonces, mi pregunta para ti es ¿por qué y cómo empezaste esa escuela? Dr. Roberto Langer. Bueno, en realidad, también, así que mi interés en la enseñanza comenzó cuando estaba en Cornell porque era asistente de enseñanza, y realmente me encanta eso. Y así, cuando vine a Boston el año siguiente y a Cambridge para unirme al MIT como estudiante de posgrado, me involucré en algunas actividades de tutoría en Rosebury y Cambridge. Dr. Roberto Langer. Y así había algunas personas, Peter Aggerty y Neil Didrikson, Paul Davies, dijeron que estaban interesados en comenzar la escuela para niños de clase trabajadora para niños, y que habían oído hablar de mí, así que me preguntaron en qué ayudé a desarrollar los programas de matemáticas y ciencias. Dr. Roberto Langer. Y pensé que esto sonaba como algo que valía la pena, así que lo hice. David Ferguson. Eso es muy bueno. Entonces, ¿cómo ha mantenido la visión y el trabajo en equipo en su entorno? ¿Cómo ha mantenido esas cosas en su entorno de trabajo, en su laboratorio? ¿Cómo mantienes esas cosas? Dr. Roberto Langer. Bueno, supongo que allí son diferentes. Con visión, trato de alentar a todos en el laboratorio a pensar en grande y soñar en grande y hacer cosas que, con suerte, podrían tener un efecto mayor en la salud y el bienestar del mundo. Dr. Roberto Langer. Entonces, ya sabes, en términos de trabajo en equipo, animo a todos a trabajar juntos. ¿Sabes? Tenemos mucha gente, tenemos más de 100 personas en el laboratorio, pero trato de contratar, quiero decir, esto puede sonar casi tonto, pero trato de contratar a personas muy agradables que se llevan bien entre sí, pero ¿qué? Dr. Roberto Langer. También que realmente quieren hacer una diferencia en el mundo, y eso es lo que he tratado de hacer repetidamente, así que creo que han sido geniales. Quiero decir, la gente que hemos tenido en nuestro laboratorio ha sido excepcional. David Ferguson. Sí, eso es muy cierto. Entonces, en términos de su éxito, creo que cualquier estándar de medición, internacional, nacional, creo que todas las personas pueden estar de acuerdo en que ha tenido éxito como pionero en el campo. Entonces, ¿qué diría que ha complementado más su éxito, ya sea la tutoría, ya sea la creación de redes, ya sea su David Ferguson? Enfoque para estudiar, aprender o enseñar, ¿cuál diría que ha sido el principal motor o impulso en el que ha tenido éxito? Dr. Roberto Langer. Bueno, creo que en la medida en que he tenido éxito, y eso podría ser discutible, pero en la medida en que lo he hecho, realmente creo que ha sido la gente. He tenido gente maravillosa trabajando en el laboratorio y han sido geniales. Dr. Roberto Langer. Me gusta pensar que se sienten, que los he tratado bien y que los he animado a intentar vivir sus sueños. Pero en realidad, es tener gente tremenda, ya sabes, en el MIT. Ha sido un gran lugar y acabamos de tener una enorme cantidad de muy buenos estudiantes y postdoctorados en el laboratorio. Dr. Roberto Langer. Y, ya sabes, por cualquier medida, ya sabes, creo que 19 están ahora en la Academia Nacional de Ingeniería. Creo que 16 además en la Academia de Medicina, ya sabes, y han recibido todo tipo de premios y han fundado empresas. Quiero decir, han tenido mucho éxito. Dr. Roberto Langer. Así que para mí, ha estado allí. David Ferguson. Si eso es verdad, las personas son importantes, muy importantes. Entonces, dadas todas sus responsabilidades y logros, ¿cómo se ha esforzado por mantener una vida equilibrada? Dr. Roberto Langer. Bueno, mi esposa es muy buena para asegurarse de que lo haga. ¿Sabes? Cuando mis hijos eran pequeños, me dijo que si no viajaba, tenía que estar en casa a las 7 todas las noches para pasar tiempo con los niños. Dr. Roberto Langer. Y cuando viajo, quiero decir, por supuesto, en este momento no estoy viajando. Pero cuando viajaba, ciertamente cuando los niños eran pequeños, quiero decir, ya sabes, incluso en casa, nunca me ausentaba por mucho tiempo. Dr. Roberto Langer. ¿Sabes? Aprendí que eso era lo importante para ellos. Entonces, para dar un ejemplo, 5 años seguidos, tuve que ir a Israel para algo como una conferencia o un premio o un título honorífico. Dr. Roberto Langer. Y todas esas 5 veces seguidas, 5 años diferentes, no usé un hotel. ¿Sabes? Acabo de volar. Pasé parte del día allí y luego volé de regreso allí. Fue entonces cuando había un vuelo de teléfono a Viva Nueva York, así que pude hacerlo. Dr. Roberto Langer. Pero, de todos modos, eso era para que nunca me fuera por mucho tiempo. David Ferguson. Está bien. Sí, la familia es una cosa muy importante. Entonces yo diría que dirías que tu familia es la base sobre la cual has podido mantener una vida equilibrada. Dr. Roberto Langer. Pues eso y mi mujer ha sido muy bueno, Laura. Ella me dice lo que piensa. Si hacía algo, si probablemente estaba pasando demasiado tiempo haciendo otras cosas, sin duda me lo haría saber. David Ferguson. Oh sí, eso es bueno. Entonces, en términos de sus impactos, sí, ¿cuáles diría que han sido sus ideas más creativas, impactantes y efectivas hasta la fecha? Dr. Roberto Langer. Bueno, creo que ha habido 3 o 4 áreas que es difícil saber, que han sido importantes. Supongo que uno ha sido el trabajo que hicimos con Judah Folkman para aislar los primeros inhibidores de la angiogénesis, los primeros inhibidores de los vasos sanguíneos, ya sabes, y eso ha llevado a mucho, debido al trabajo que otros habían hecho. Dr. Roberto Langer. Después de eso, las compañías y demás, los muchos medicamentos nuevos que serían útiles para tratar el cáncer y las enfermedades de los ojos. El segundo es probablemente el trabajo sobre nuevos materiales y la administración controlada de fármacos, que también incluye la nanotecnología. Dr. Roberto Langer. Y eso también llevó, ya sabes, a abandonar una serie de productos de diferentes grupos nuevamente. Y finalmente sería la idea de eso con el concepto de ingeniería de tejidos. Ya sabes, donde podrías combinar materiales y células y, ya sabes, crear nuevos tejidos y órganos, ya sea un órgano en un chip o un nuevo tejido en el cuerpo, como piel artificial. David Ferguson. De acuerdo. Entonces, en términos de encontrar y buscar el entorno adecuado para que prosperes científica e intelectualmente, ¿cómo haces para encontrar eso? ¿Cómo y cuál fue tu guía o cuál fue tu rúbrica o base en la que encontraste ese ambiente? David Ferguson. Supongo que fue el MIT. Pero, ¿cómo encontraste eso? Dr. Roberto Langer. Sí, bueno, quiero decir, hay diferentes maneras. Quiero decir, realmente, supongo que la experiencia que tuve en el Children's Hospital fue realmente clave. Y luego MIT, quiero decir, ya sabes, eso fue, supongo, buena suerte que tuve, pero sé que me contrataron y, ya sabes, las cosas funcionaron allí. Dr. Roberto Langer. En última instancia, sin embargo, realmente creas tu propio entorno. Cuando ejecuta un laboratorio, ya sabe, es el entorno que crea para los demás. Ya sabes, eso termina marcando la diferencia que termina marcando la diferencia. David Ferguson. Sí, es interesante que digas eso porque he escuchado a otras personas decir que, bueno, no se trata tanto de que el entorno ya esté allí, sino que puedes complementarlo para crear el entorno por ti mismo. Dr. Roberto Langer. Creo que esa es la clave. Sí, estoy de acuerdo. Estoy de acuerdo con él. David Ferguson. Sí, esa es la clave para poder prosperar científica e intelectualmente. Escuché a alguien decir que casi puedes modificar el entorno y complementar el cambio, ¿verdad? Dr. Roberto Langer. Entonces, tienes un gran efecto en el medio ambiente, por la forma en que tratas a las personas y la forma en que actúas como un modelo a seguir o la falta de él. Eso es cierto. David Ferguson. Eso es muy cierto, tus acciones sí cuentan. Entonces, en términos de usted, manteniendo una visión del panorama general en su carrera y su vida en general, ¿cómo lo ha hecho, cuando enfrentó desafíos u obstáculos? David Ferguson. ¿Cómo ha mantenido una visión del panorama general? ¿Qué te mantuvo en marcha y qué te mantuvo persistente? ¿Cómo hiciste eso? Dr. Roberto Langer. Bueno, supongo que solo soy yo, ya sabes, soy una persona muy terca. Si creo en algo, sigo intentando y tratando. No me rindo fácilmente si siento que algo puede hacer algo bueno. Si siento que hemos hecho un hallazgo, eso es importante. Dr. Roberto Langer. Ya sabes, lo haré. Haré lo mejor que pueda. David Ferguson. De acuerdo. Sí, eso es muy bueno. Entonces, Dr. Langer, ya sabe, mi trayectoria profesional, hablo sobre mi trayectoria profesional. Eso es lo que comencé como ingeniería en Georgia Tech, y sucedieron algunas cosas que me llevaron a hacer la transición a otra institución y cambiar mi especialización a bioquímica. David Ferguson. Entonces cambié de camino porque mis intereses también cambiaron. Entonces, mi pregunta para ustedes es para aquellos en el campo de la ingeniería, ¿cuáles serían algunos de sus consejos o cuáles serían las cosas clave que hicieron, que los complementaron para poder hacer la transición a un programa de doctorado, un programa de ingeniería, eventualmente y luego servir como pionero en el campo? ¿Cuáles fueron algunas de las cosas clave que hizo en su tiempo como estudiante universitario, otro estudiante de posgrado? Dr. Roberto Langer. Sí, bueno, creo que como estudiante universitario, siento que realmente quieres aprender los fundamentos y realmente quieres que te vaya bien en clase. Quiero decir, y esa debería ser tu máxima prioridad. Dr. Roberto Langer. Quiero decir, no haría investigación u otras cosas hasta que supiera que podría hacerlo bien en clase, por así decirlo, como estudiante de posgrado, creo que entonces, ya sabes, también quieres hacer lo mismo, continuar aprendiendo el fundamentos bien, pero luego realmente desea elegir un proyecto de investigación y un asesor de investigación. Dr. Roberto Langer. Si estás obteniendo un doctorado o incluso un título de maestría, que es realmente un buen mentor. ¿Y qué significa eso? Es decir, ojalá que te traten bien y que te pongan en un ambiente o te tengan en un ambiente donde puedas aprender de ellos y de los demás. David Ferguson. De acuerdo. Sí, eso está bien. Así que creo que la mayoría de nosotros podemos estar de acuerdo en que esta vez ha sido un tanto desafiante para mucha gente debido a la pandemia y mucha gente está encontrando obstáculos y desafíos. ¿Qué te ha mantenido optimista durante este tiempo, durante esta pandemia? Dr. Roberto Langer. Bueno, dos cosas. Una de las cosas que he estado haciendo es que he pasado mucho tiempo tratando de luchar contra esta pandemia. Ya sabes, una de las empresas que ayudé a iniciar es Moderna, ha sido una de las empresas líderes en términos de creación de una vacuna contra el COVID, ya sabes, por lo que ahora están en lo que se llama ensayos de fase 3 para hacerlo. Y, casi todas las compañías que están haciendo una vacuna de ARN y que son las líderes en términos de avanzar en la clínica, usan nanopartículas, ya sabes, que se usan para administrar el ARN. Dr. Roberto Langer. Y, ya sabes, esa es un área en la que hemos hecho mucho. Y también estamos trabajando en el laboratorio sobre vacunas con máscaras, creando nuevas máscaras, creando nuevos ventiladores, creando ventiladores divididos, ya sabes, trabajando en aerosoles nasales que podrían combatirlo. Dr. Roberto Langer. Estamos trabajando en nuevos análisis de sangre, así que hemos estado muy activos en términos de realmente tratar de hacer una diferencia en el tratamiento de COVID, además de, ya sabes, a pesar de que la investigación se desaceleró en otras cosas debido a las reglas en las universidades. Dr. Roberto Langer. ¿Sabes? Creo que continuamos esa investigación también, y estamos trabajando mucho con la Fundación de X en cosas que pueden ayudar al mundo en desarrollo, y solo otras cosas que espero que sean importantes para el mundo en algún momento. Dr. Roberto Langer. Y de nuevo, creo en todas estas cosas, así que no vamos a parar, ¿sabes? Y así, pero todo eso es lo que hemos tratado de hacer. David Ferguson. Entonces, tengo dos preguntas más en términos de su trabajo con la pandemia de COVID-19, en términos de todas las cosas que está diseñando e ingeniando. ¿Has visto el impacto o todavía está en proceso? David Ferguson. ¿Has comenzado a ver impactos de tu trabajo hasta ahora? Dr. Roberto Langer. Diría que la respuesta a eso es que hemos visto algún impacto, quiero decir, entonces, ¿qué puedo decir? El hecho de que me refiero a algunas de las cosas que hicimos ahora hace más de 45 años, creando, ya sabes, pequeñas partículas que podrían liberar ácidos nucleicos. Dr. Roberto Langer. Quiero decir, como digo, lo están utilizando Moderna, Pfizer, muchas empresas. Quiero decir, de nuevo, muchas otras personas contribuyeron, pero lo hicimos temprano. También desarrollamos otros tipos de nanopartículas. Una y otra vez, mucho de esto lo están haciendo nuestros estudiantes y algunos de estos lugares. Dr. Roberto Langer. Así que creo que las vacunas aún no han tenido un impacto. Pero aparte de eso, las personas que han sido tratadas por él. Pero me parece que la esperanza más grande de la nación, la esperanza más grande del mundo es que las vacunas son las cosas que hemos hecho. Dr. Roberto Langer. Ayudamos a hacer nuevas máscaras. Y ya sabes, varios millones de esos ya han sido utilizados por personas. Y creo, ya sabes, hay mejores máscaras, ya sabes, así que algunas de las otras están en proceso y están en ensayos clínicos. Dr. Roberto Langer. Y, ya sabes, se han movido muy rápido. Entonces, así que creo que ya hemos podido tener algún impacto en los pacientes y, con suerte, más. David Ferguson. Entonces, ya sabes, mencionaste, volver, mencionaste cómo dijiste que eres algo terco o que eres persistente. ¿Es esto algo que te enseñaron en tu crianza o es algo que desarrollaste con el tiempo en entornos académicos? Dr. Roberto Langer. Creo que es algo que simplemente nací así, ya sabes, creo que siempre he sido terco. Estoy seguro, mis padres pensaron eso. David Ferguson. Ah, ok, sí, bueno, Dr. Langer, fue tan bueno tenerlo hoy. Realmente aprecio que se haya tomado el tiempo para conversar conmigo y que discutamos su trayectoria profesional. Y actualmente está marcando una diferencia sustancial en los campos de la ingeniería química y la ingeniería biomolecular. Dr. Roberto Langer. Gracias. Es un placer y mucha suerte. Esta entrevista se grabó en 2020 para The New Chemists Podcast, y fue producida y presentada por David Joshua Ferguson. Ferguson.
Welcome to the New Chemists. We're glad you're listening. Feel free to download this podcast on Apple Podcasts, Google Podcasts, and Spotify. Here on the New Chemist.
Didn't have a map, just a pencil in my hand. Good at science, good at math, trying to understand. They said be an engineer, so I...
Community, research, and COVID-19. We're happy you're tuning in. My guest today is Dr. Robert Langer. Thanks for joining me today. It is good to hear from you.
Followed that advice, found a spark in a chemistry class. Didn't have to think twice, wasn't all planned out, just a search for something new. Helping start a school for kids with a different point of view. I meant to live the words that he would never have to say. He showed me that the biggest walls can sometimes melt away. He said, go on and dream those big dreams, paint a whole new...
Inventor and one of the 12 Institute professors at the Massachusetts Institute of Technology. He was formerly the Germershausen Professor of Chemical and Biomedical Engineering and
shade of sky. Believe that anything is possible, then give it a real try. Cause you're gonna find the roadblocks, you're gonna hear the doubt, but that stubborn beating in your heart is what it's all about. So don't you give up, no, don't you give up now.
of Biological Engineering at MIT. He is also a faculty member of the Harvard MIT Program in Health Sciences and Technology and the Koch Institute.
It's easy doing incremental, adding piece by piece, but a transformative idea gives the world a new release. So I try to hire nice people who want to make a change, build a team that works together to rearrange the page. From nanoparticles fighting a pandemic's awful sting, to tissue engineering the new life that we can bring.
Cancer research. In 2015, Dr. Robert Langer was awarded.
It's more than just the accolades or prizes on a shelf. It's the environment you're building, bigger than yourself. Remember, go on and dream those big dreams. Paint a whole new shade of sky. 'cause you're gonna find the road.
Welcome, Dr. Robert Langer.
Blocks, you're gonna hear the doubt, but the stubborn beating in your heart is what it's all about. So don't you give up. No, don't you give up now. My wife says be home by 7, keep a balanced point of view. Never be gone for too long from the ones who count on you. It's the bigger picture calling through the static and
Okay, Dr. Langer, thank you so much for joining me today. It is good to have you here.
the noise. It's the people you empower in their triumphs and their joy.
To do things that can hopefully have a big effect on the health and well-being of the world. And so, you know, in terms of teamwork,
Didn't have a map, just a pencil in my hand. Good at science, good at math, trying to understand. They said be an engineer, so I followed that advice. Found a spark in a chemistry class, didn't have to think twice. Wasn't all planned out, just a search for something new. Helping start a school for kids with a different point of view. I meant to live the words that he would never have to say. He showed me that the biggest walls
encourage everybody to work together. You know, we have a lot of people. We have over 100 people in the lab. But I try to hire, I mean, this may sound almost silly, but I try to hire very nice
and sometimes melt away. He said, go on and dream those big dreams. Paint a whole new shade of sky. Believe that anything is possible and give it a real try. 'Cause you're gonna find the roadblocks, you're gonna hear the doubt, but that stubborn beating in your heart is what it's all about. So don't you give up. No, don't you give up now. It's easy doing incremental, adding piece by piece, but a transformed idea gives the world a new release. So I try to hire nice people who want to,
but who also really want to make a difference in the world. And so that's what I've tried to do repeatedly. And they've been great. I mean, the people we've had in our lab have been outstanding.
Yeah.
make a change, build a team that works together to rearrange the page. From nanoparticles fighting the pandemic's awful sting, to tissue engineering the new life that we can bring. It's more than just the accolades or prizes on a shelf. It's the environment you're building, bigger than yourself. Remember, go on and dream those big dreams. Paint a whole new shade of sky. Believe that anything is possible and give it a real try. 'Cause you're gonna find the roadblocks, you're gonna hear the doubt, but the stubborn beating in your heart is what it's all about.
In terms of your success, I think by any standard of measurement, international, national, I think people can all agree that you have been successful as a pioneer in the field.
Don't you give up. No, don't you give up now. My wife says be home by 7, keep a balanced point of view. Never be gone for too long from the ones who count on you. It's the bigger picture calling through the static and the noise. It's the people you empower in their triumphs and their joy.
about the environment being there already, but you can complement to creating that environment for yourself.
I think that's key, yes.
Didn't have a map, just a pencil in my hand. Good at science, good at math, trying to understand. They said be an engineer, so I followed that advice. Found a spark in a chemistry class, didn't have to think twice. Wasn't all planned out, just a search for something new. Helping start a school for kids with a different point of view. I meant to live the words that he would never have to say. He showed me that the biggest walls
able to thrive scientific intervention. I heard someone say you can almost somewhat bend the environment to your will or complement it, change.
And sometimes melt away, he said, go on and dream those big dreams. Paint a whole new shade of sky. Believe that anything is possible and give it a real try. 'Cause you're gonna that stubborn beating in your heart is what it's all about. So don't you give up. No, don't you give up now. It's easy doing incremental, adding piece by piece, but a transformative idea gives the world a new release. So I try to hire nice people who want to.
If you have a big effect on the environment by the way you treat people and the way you act as a role model or lack thereof.
That's true. It's very big reaction.
Make a change, build a team that works together to rearrange the page. From nanoparticles fighting a pandemic's awful sting, to tissue engineering the new life that we can bring. It's more than just the accolades or prizes on a shelf. It's the environment you're building, bigger than yourself. Remember, go on and dream those big dreams. Paint a whole new shade of sky. Believe that anything is possible and give it a real try. 'Cause you're gonna find the roadblocks, you're gonna hear the doubt, but that stubborn beating in your heart is what it's all about.
In terms of you maintaining the view of the bigger picture in your career and in your life in general, how have you done that? When you face challenges or obstacles?
Don't you give up. No, don't you give up now. My wife says be home by 7, keep a balanced point of view. Never be gone for too long from the ones who count on you. It's the bigger picture calling through the static and the noise. It's the people you empower in their triumphs and their joy.
Purpose of these episodes not at all for advice or medical suggestions. These are aimed to provide support to peer pharmacists in training in educational and intellectually stimulating ways. Again, these are not at all for medical advice or medical suggestions. Please see your local board and state-certified health professional. The views of this podcast represent those of my guests and I. Purpose of these episodes not at all for advice or medical suggestions. These are aimed to provide support to peer pharmacists in training in educational and intellectually stimulating ways. Again, these are not at all for medical advice or medical suggestions. Please see your local board and state-certified health professional. The views of this podcast represent those of my guests and I. Purpose of these episodes not at all for advice or medical suggestions. These are aimed to provide support to peer pharmacists in training in educational and intellectually stimulating ways. Again, these are not at all for medical advice or medical suggestions. Please see your local board and state-certified health professional. The views of this podcast represent those of my guests and I.
Welcome to the debate. Our discussion today, well, it centers on one of the most critical challenges in oncology. How do we accurately interpret patterns in cancer therapy response to really achieve true precision medicine?
Right. The core mission is always matching the patient to the most effective treatment. Absolutely. But how we define that effectiveness, and maybe more importantly, what variables we prioritize in our research models, that's where the real, intellectual friction lies. That's where progress can either accelerate or stall.
Exactly. We're sort of questioning the very foundation of predictive modeling here. The central question for us today is, should oncology research in clinical trial design systematically integrate embryonic lineage, ectoderm, mesoderm, endoderm, as a fundamental contextual variable? Should this sit alongside the established methods like molecular subtype and microenvironmental profiling that we currently rely on?
And, I'm going to argue that introducing such a broad, almost historical classification, it's not just potentially an inefficient use of limited research resources. I think it actually risks actively diverting focus, introducing noise, and ultimately slowing down the kind of high-resolution molecular targeting that's given us our most successful recent therapeutic advances. That's where I see the real progress being hampered.
And I'm taking the opposing view. My stance is that integrating this developmental logic is essential for foundational progress. It targets the most fundamental, stable cellular behavior, the corrupted developmental state itself. And this provides a necessary sort of systems-level map to contain what can otherwise seem like overwhelming molecular chaos. Progress, from my perspective, needs this broader context.
Okay, that sets the stage pretty clearly. Now, if we're really talking about progress, we absolutely have to look at what delivers actionable results. Today, I can see why you think that developmental history might define the future path, but let me maybe offer a different perspective on where true, reliable progress seems to be coming from right now. You're arguing for looking backward, at the origin. I'm arguing we need to look precisely at the current acquired molecular machinery that's actually driving the cancer now.
Well, my position is that true progress really requires understanding cancer not just as a random collection of accumulated mutations, but as a deeply and systematically corrupted developmental process. Think about the hallmark of phenotypic plasticity. This is the tumor cell's ability to evade terminal differentiation, to, de-differentiate back to these progenitor-like states, or maybe to short-circuit the differentiation pathway entirely. This plasticity, I'd argue, is a core acquired hallmark capability. And its rules, its constraints, are fundamentally written by the cell's lineage.
Okay, we can agree plasticity is a key hallmark for survival, no question. But does the initial germ layer, ectoderm, mesoderm, does that truly determine how that plasticity gets used years later after the cell has picked up dozens, maybe hundreds of key mutations? The challenge right now, and the source material really emphasizes this, is navigating the sheer, immense complexity of all these interlinked signaling pathways.
Exactly. That complexity is precisely why we need an organizing principle. The material talks about the need to define comprehensive maps in cancer signaling networks. Lineage, I argue, provides that macro-level context. It defines the inherent biological constraints and possibilities for the molecular changes that follow. Now, to illustrate the therapeutic potential here, let's consider acute promyelocytic leukemia, APL.
A specific blood cancer, right? Go on.
Yes, it's a specific, quite aggressive form of myeloid leukemia where the myeloid progenitor cells, these are cells destined to become certain white blood cells. They get stuck. They get stuck in a proliferative, sort of immature stage. Now, successful treatment involves coercing those cells, basically forcing them to undergo terminal differentiation using retinoids. This makes the cells stop dividing and enter a permanent post-mitotic state. That's a direct mechanistic proof that the developmental state itself is an actionable therapeutic target. This isn't just about killing the cell, it's commanding its stable foundational state. that feels like fundamental progress.
Sorry, but I just don't quite buy that the APL model is generalizable proof for integrating these really broad concepts like ectoderm or mesoderm into designing trials for complex solid tumors. APL, well, it's a liquid cancer, right? It's typically driven by one defining translocation, a very specific molecular event, the PML-RAR alpha fusion, and the differentiation therapy is highly targeted to that specific blocked maturation pathway. It's elegant, yes, but is it widely applicable for making progress elsewhere?
But the principle is systems level. It shows you can target the state, not just the mutation.
I guess I just come at it from a different angle. For me, focusing on broad, coarse-grained variables like embryonic lineage feels like a conceptual step backward, especially when precision medicine is so clearly moving toward finer and finer-grained molecular profiling. Progress really hinges on identifying specific individual genetic and epigenetic alterations. Cancer is inherently a multi-gene, multi-step disease, starting from, a single abnormal cell. We absolutely must follow the molecular trail, the one that's active now, not necessarily the historical one laid down during embryogenesis.
But why must we view lineage simply as a historical footnote? Why not see it as the stable foundation upon which that molecular trail is inevitably laid? Don't the foundation rules affect the trail?
Because the challenges we're facing, the ones holding back progress now, are contemporary and dynamic. We're already struggling to integrate the local, specific variables. The source material highlights this immense complexity of, say, non-mutational epigenetic reprogramming like wholesale changes in the methylome. These chemical tags on DNA, driven by localized factors like chronic hypoxia or inflammation, and then there's the influence of highly individual polymorphic microbiomes. All of these factors are immediate, highly specific, often localized, and arguably far more relevant to predicting treatment response today than the originating germ layer from development.
But surely these dynamic processes still exist within a biologically constrained context set by that origin.
Yes, a context exists, but progress, I believe, is best achieved by completing the, quote, complete integrated circuit of the cancer cell, and we do that through routine, high-throughput expression profile analysis of genetic mutations and epigenetic modifiers. We need that full molecular map first. Just inserting a broad lineage classification before that detailed profiling, well, it doesn't seem to enhance the precision we need. It potentially muddles the signal, forcing us into these macro categories, when the solution, the path to progress, likely lies in the micro mechanisms.
Okay, let's try to ground this in specific tumor behavior. Let's focus on a key hallmark that really distinguishes cancers: metastasis. The ability to induce and sustain hallmark capabilities like tissue invasion and metastasis differs vastly by tumor type and tissue environment. Lineage, I propose, provides an inherent organizing framework for these behaviors that just focusing on general molecular targeting often misses.
How so, specific?
Well, consider the morphological changes required for successful metastasis. Cells often need to undergo something like an epithelial to mesenchymal transition, or EMT. This requires relying on specific adhesion molecules, things like EEC adherin, and changes in integrin expression. Now, a carcinoma, which originates from ectoderm or endoderm epithelial cells, it uses one set of regulatory circuits to shed its epithelial identity and invade. A sarcomoma, on the other hand, derived from the mesoderm connective tissues, it might have a fundamentally different, often more aggressive inherent migratory capability simply because its progenitor cell was already loosely adherent. So the regulatory circuits governing, say, the loss of EEC adherin function and the switch in integrin expression, they are almost certainly lineage dependent, rooted in the initial cell of origin's developmental program. If we know the lineage, perhaps we know something fundamental about the inherent resistance or susceptibility to invasion, which is, of course, a major factor in treatment failure and lack of progress in cures.
Okay, that's a compelling argument based on tissue type. I'll grant you that. But have you considered that the fundamental hallmarks of cancer, things like sustained growth signals, avoiding apoptosis, tissue invasion, these are capabilities acquired via mutations in universal regulatory components? We're talking about critical proteins like P53 ARB or general disruption of ubiquitous growth factor receptor pathways like EGFR. These are common across many cancer types.
Agreed. They are common targets.
Right. So while the exact regulatory circuit driving the metastasis phenotype might differ between a sarcoma and a carcinoma, progress in therapy, historically and currently, is made by targeting the final common pathways. For instance, targeting VEGF for angiogenesis or developing inhibitors for specific oncogenic fusion proteins like BCR-ABL.
Maybe just briefly clarify BCR-ABL for our listeners?
Oh, sure. BCR-ABL is sort of the classic example of a rogue enzyme. It's a fusion protein created when two genes improperly join together. This single constitutively active enzyme drives the proliferation of chronic myeloid leukemia, or CML. And the incredibly successful drug, imatinib, targets this enzyme specifically. My point is, regardless of the cell's distant developmental ancestry, the focus for making progress has been on the acquired mutations that drive functional autonomy. The developmental history in this view is sort of overshadowed by the acquired pathology that we can directly target.
I agree, we absolutely must target the machinery. But my point is the developmental program dictates the rules of engagement for that machinery. A mutation in P53, for example, it surely acts differently depending on the cell type it resides in and the inherent plasticity granted by its germ layer origin. The context matters.
Look, for practical, actionable progress in clinical settings, high specificity wins almost every time. Just look at the immense success we've achieved with highly specific drug regimens tailored by molecular profiling analysis. This type of molecular precision dramatically improves patient outcomes and, crucially, minimizes the nonspecific toxicity inherent in earlier generalized treatments like old-school chemo. Adding a broad lineage filter before you do the molecular profiling just feels like an unnecessary step. It risks complicating patient stratification in trials, maybe requiring larger cohorts, and potentially delaying the patient from getting to the truly precise treatment dictated by their specific gene signature. To me, that impedes progress. It doesn't accelerate it.
I'm just not convinced by that line of reasoning, primarily because focusing only on current molecular targets fails spectacularly when tumors are heterogeneous or, critically, when they become drug-resistant by, as the source material puts it, continually changing their makeup. Genomic instability is itself a core hallmark of cancer. That means relying solely on a single specific mutation profile is inherently vulnerable to adaptive resistance. That's a huge barrier to long-term progress.
But that instability is exactly why we need rapid, high-resolution profiling to keep up. We have to monitor the moving target.
Yes, but maybe a strategy based on developmental logic, like differentiation therapy, forcing the cell into a stable, non-proliferative state, attacks the tumor's fundamental, foundational stable state. It offers a robust, perhaps distinct therapeutic angle that might overcome resistance pathways driven by that very molecular instability. It's about establishing a therapeutic vulnerability that's distinct from the genetic lesions themselves. And furthermore, think about a systemic target like angiogenesis. Anti-angiogenesis agents, they target the microvasculature endothelial cells. Now, these cells are genetically more stable than the tumor cells they feed. A developmental, maybe systemic approach could help identify these more stable regulatory targets, which seems crucial for long-term progress against such an unstable opponent.
That's an interesting point about targeting stable components. Though I might frame the overall danger differently. The danger, in my view, isn't really simplification per se. The fact is, the molecular and microenvironmental landscape is already overwhelmingly complex. We are already dealing with non-mutational epigenetic reprogramming, the modulation of immune responses by highly polymorphic microbiomes, the unique collection of microbes in a patient's gut or even within the tumor, which can vary radically person to person.
And why does that complexity argue against considering lineage?
Because it argues for specificity. The microbiome, for instance, creates highly localized, specific pressures that impact everything from drug metabolism to immune surveillance. Introducing a third, potentially less precise, broad biological variable like embryonic lineage into trial design, well, it just seems likely to introduce significant noise into the analysis.
It complicates the already difficult statistical task of identifying the truly influential genomic and microenvironmental factors that our most advanced technologies are already cataloging. We need to define constraints, yes, but constraints that lead to highly specific therapeutic avenues, not just more statistical variables that slow down progress.
But lineage could help make that complexity manageable by defining the potential constraints on how cells can mutate and where they can go pathologically. It sets the stage, maybe defines the geometry of the disease in a way. Without that systemic map, we risk having these islands of perfect molecular data without really knowing how they fit into the overall geography of the cancer process. The danger of relying solely on pure molecular simplification is precisely what the source material warns against, that it, quote, reflects little of the biological reality of cancer progression in human patients in vivo.
And the danger, as I see it, of incorporating a broad historical variable like germ layer origin is sacrificing the necessary molecular granularity required for modern clinical action and, frankly, for regulatory approval of new drugs. We have to prioritize factors that offer the highest degree of predictive value in the immediate term, leading to tangible progress like new specific drug approvals.
So to conclude my side, I'd already assert that incorporating developmental logic is, I think, a necessary evolution. It aligns research with the ultimate truth that cancer is fundamentally a corrupted developmental entity. It pushes oncology towards a more comprehensive systems-level understanding. Real, lasting progress requires addressing the root state, things like plasticity and differentiation blockage, not just the downstream symptoms of mutation and gene expression changes.
And my position remains, well, skeptical. While germ layer origin is certainly biologically meaningful, I'm still unconvinced it provides superior predictive or therapeutic value compared to the rapid advances in molecular, genetic, and epigenetic profiling that we are currently leveraging. Progress, for me, must remain ruthlessly focused on the most actionable and precise molecular mechanisms driving the disease in the individual patient right now.
It's clear, though, that the quest for precision in oncology demands that we constantly seek new organizing principles, whether they stem from the patient's deep developmental history or their immediate molecular fate.
Indeed. And the ultimate success, I suppose, lies in continued rigorous challenge and integration, or sometimes rejection, of these multiple perspectives. Always driven by the empirical evidence that shows where we should truly focus our resources to make the most effective progress.
Today, we are tackling, well, a really immense challenge: cancer. It's not just one thing, is it? It's this umbrella term for, gosh, over 200 diseases, all defined by this fundamental breakdown, uncontrolled growth, and crucially, the ability to spread.
It really is. It's like the ultimate biological rebellion where the cell's own rule book, its genome, gets rewritten. And that disrupts this incredibly delicate balance, the one between cells dividing when they should and staying quiet when they should.
A delicate balance indeed.
So our goal here in this deep dive is to take these complex sources you've gathered on cancer biology, everything from the basic molecular nuts and bolts of how a cell goes wrong, right up to the cutting-edge research that's trying to fight back and really pull out the key insights for you.
It's a fascinating, if daunting, area, a real cellular chess game. But before we get into the strategies of that rebellion, we absolutely have to include our standard and very important disclaimer for you, the listener.
Yes, absolutely crucial. Please note the views of this discussion reflect those of my colleague and I, and AI may have been used in the production of the episode. We are deep diving into scientific research and development, not offering clinical management advice.
That's key.
Please see a board-certified medical professional, one that is state-licensed, for medical or professional advice regarding any health concerns. We disclaim any loss in any way regarding the application of this information.
Okay, thank you for that. Right then, let's unpack this. If cancer starts with the genome, we need to go right back to the beginning. How does a single solitary cell out of, well, trillions, actually manage to go rogue?
Well, the central idea, the core dogma really, is that cancer isn't usually a sudden, widespread event. It's described as a multi-gene, multi-step disease. And it nearly always starts from just one single abnormal cell. That's what we call its clonal origin.
I don't know origin.
Right. This cell has picked up some change in its DNA sequence, a mutation that kicks things off.
So it's not like a whole tissue fails at once. It's one bad apple, initially, that then starts a kind of internal evolution.
Precisely. That's a great way to put it. That single cell leads to what's called clonal expansion. First, you get this uncontrolled division, just making more and more copies of that abnormal cell. But it doesn't stop there. The resulting mass of cells isn't uniform.
Okay.
You then get successive rounds of new mutations happening within that growing population, and then this is key selective expansion. Those cells that happen to acquire mutations, giving them an advantage.
An advantage for the cancer, you mean, like dividing faster.
Exactly. Faster division, better survival, ability to resist signals telling them to die, whatever gives them a competitive edge over their neighbors, they start to dominate. It's pure Darwinian selection, but happening inside the body at the cellular level. And eventually, this leads to a detectable tumor.
And if you catch it then, it might just sit there, maybe benign. But what pushes it over the edge to become, well, lethal?
Yeah, the lethal outcome usually comes down to the tumor's ability to invade surrounding tissues, erode normal structures, and critically, to spread. That process is metastasis, spreading to other vital organs. That's the end game of this uncontrolled clonal expansion cells breaking out, colonizing new territories, and causing organ failure. That's what causes death in most cases.
It seems like there should be built-in limits, though. A cell designed to divide can't just go on forever, surely. I mean, normal cells, healthy ones, they have a kind of counter, don't they? They divide maybe 50, 60 times and then just stop senescence.
That's right. That's the Hayflick limit. It's a fundamental safeguard baked into our normal somatic cells, the non-reproductive ones. So for cancer to succeed long-term, it absolutely has to find a way around this limit. It needs what we call limitless replicative potential. Cellular immortality, essentially.
How on earth does it achieve that?
They figure out how to manipulate a very specific enzyme, telomerase.
Ah, telomeres. I always think of them like the little plastic tips on shoelaces protecting the ends of the chromosomes.
That's a perfect analogy. Telomeres are these repetitive DNA sequences at the very ends of our chromosomes. They don't code for proteins, they're just protective caps. But every time a normal cell divides, a little piece of that telomere gets lost, it shortens. Eventually, it gets so short the cell senses damage and triggers senescence or cell death.
So the shoelace unravels, basically.
Kind of, yeah. But cancer cells manage to switch on or ramp up the production of telomerase. This enzyme is a reverse transcriptase. It actually rebuilds those repetitive DNA sequences, adding them back onto the ends of the telomeres. So by constantly repairing the shoelace tips, they prevent that shortening, bypass the Hayflick limit, and effectively become immortal. They can just keep dividing.
Immortality is one thing, quite a big thing actually, but the cell still has to deal with all the signals telling it when to divide and when to stop. You mentioned disrupting the balance. This feels like we're getting into the core hallmarks, messing with the signaling pathways, turning the go button on permanently and breaking the stop button.
Absolutely spot on. You have to tackle both. Let's start with the go signals, the ones driven by oncogenes. Normally, a cell is quite dependent. It needs signals from outside, often from different cell types, what we call heterotypic signals, just to get the green light to divide.
But cancer cells become independent. They don't need outside permission.
Precisely. They often develop what's called autocrine stimulation.
They make their own go signals.
Yeah, they start producing the very growth factors that are normally made by other cells, but they make them for themselves. They essentially create this feedback loop where they stimulate their own growth, drastically cutting down their need for external cues.
Is there a good example of that?
Glioblastoma, a type of brain cancer, is a classic one. These cells can learn to produce something called platelet-derived growth factor, or PDGF. Now, normally, PDGF tells other cells to grow, but if these glioblastoma cells start making themselves, maybe triggered by an oncogene like VSES, they release it, and it binds right back onto their own PDGF receptors on their surface.
Creating a constant grow now loop.
Exactly, a constitutive, always-on growth signal. It's a massive strategic advantage for the cancer cell. Self-sufficiency.
Okay, so that's jamming the accelerator down. What about disabling the brakes? The anti-growth signals, typically managed by tumor suppressor genes. These usually push cells into, a quiet G0 phase or make them differentiate permanently.
That's right. And the fascinating thing is that many of these different anti-growth pathways seem to converge on one crucial protein, the retinoblastoma protein, or PRB.
PRB.
The gatekeeper.
It acts like a central gatekeeper for the cell cycle, yes. And cancer needs to disable it. Here's how it often works. In a healthy non-dividing cell, PRB is mostly unphosphorylated. It doesn't have phosphate groups attached. In this state, it grabs hold of another set of proteins called E2F transcription factors.
And E2F normally turns on genes needed for division.
Exactly. Genes needed for DNA replication moving into the S phase of the cell cycle.
Yeah.
So by holding onto E2F, PRB keeps those genes switched off. It enforces the stop signal.
So how does cancer get past that? The cell cycle itself is pushed forward by enzymes called cyclin-dependent kinases, or CDKs. Cancer cells often find ways to crank up the activity of specific CDKs, like the cyclin DCDK46 complex. These activated CDKs then phosphorylate PRB. They stick phosphate groups onto it, lots of them. It's called hyperphosphorylation.
And it changes PRB somehow.
It changes its shape, its conformation. This makes PRB let go of E2F. Once E2F is free, it heads straight to the DNA and switches on all those proliferation genes. The cell cycle engine kicks into gear, often without any proper external signal telling it to do so. The brakes are off.
And I remember reading that viruses are particularly good at messing with PRB.
Oh, absolutely. Viruses are masters of manipulating cellular machinery. A prime example is the human papillomavirus, HPV. Its E7 aquaprotein, which is strongly linked to cervical cancer, physically binds directly to PRB. It essentially sequesters it, pulls it out of action, achieving the same result as hyperphosphorylation E2F gets released, driving uncontrolled growth. It beautifully shows how genetics, signaling, viruses, they can all converge on these key control points.
Okay, so PRB is the gatekeeper for division. But there's another really famous one, often called the guardian of the genome, P53. It sounds like the ultimate failsafe.
P53 is arguably one of the most critical tumor suppressors we have. Its role is absolutely central. When a cell experiences serious stress, think DNA damage from radiation or chemicals, lack of oxygen, hypoxia, even the inappropriate activation of an oncogene P53 steps up.
What does it do?
It gets stabilized and activated. As a transcription factor, it then switches on a whole suite of target genes to try and deal with the crisis. Depending on the situation, it might trigger cell cycle arrest hitting the pause button, often by activating a gene called P21 to give the cell time to repair the damage.
Ooh.
Or it can directly activate DNA repair pathways. But if the damage is too severe, if it's beyond repair, P53 makes the ultimate call. It initiates apoptosis, programs cell death, essentially telling the damaged cell to commit suicide for the good of the organism.
Wow. So P53 is policing DNA integrity, controlling the cell cycle, and acting as judge, jury, and executioner. Cancer must find a way to get rid of it to survive, especially if it's accumulating mutations.
It's almost a prerequisite, and the statistics bear this out. P53 is the single most commonly mutated gene in human cancers. Something like close to 50% of all cancers have mutations in the P53 gene. And in some really aggressive types, like squamous cell carcinoma, that figure can jump up to 80% or even higher.
And losing P53 means?
It means the cell loses its primary mechanism for sensing and responding to DNA damage. So even with a genome that's falling apart, riddled with mutations, the cell just keeps dividing. There's no break, no repair trigger, no suicide command. Often, the mutations are what we call dominant negative. This means that even if the cell still has one good copy of the P53 gene, the protein made by the mutated copy can actually interfere with and inactivate the remaining good protein. It's a very efficient way for the cancer cell to completely dismantle its own safety system. So for a long time, the story really centered on those core genetic hits, activating oncogenes, inactivating tumor suppressors like PRB and P53. But, and this is where things get really fascinating in recent years, the picture is broadening considerably. We're now appreciating things like cell plasticity, changes that aren't mutations, and the huge role of the environment around the tumor.
Okay, let's dive into that. First up, phenotypic plasticity. We talked about terminal differentiation being the normal endpoint, right? Cells mature, stop dividing, do their job. Cancer needs to break that rule.
Exactly. Phenotypic plasticity is basically the cancer cell's ability to dodge or even reverse that final differentiated state. It's a crucial survival skill because it lets the cell regain or retain the ability to proliferate, which differentiation normally shuts down permanently.
And the sources mentioned three main ways this plasticity shows up. The first one was dedifferentiation.
Right. Dedifferentiation is like hitting the rewind button. You have a mature, specialized cell that somehow reverts back to a more primitive, progenitor-like state. A good example is in advanced colon cancers. They can lose certain factors like HOXA5 and SM84 that normally maintain their differentiated identity. Losing these allows them to sort of demature, become more like stem cells again, regaining that critical ability to divide and proliferate.
Okay, becoming younger to keep dividing. The second one was blocked differentiation. That sounds almost like getting stuck in the worst possible phase.
It is, from the body's perspective. Here, you have progenitor cells that are supposed to mature into specific non-dividing types, but they get trapped. Regulatory changes essentially freeze them in that proliferative immature state. They just keep dividing instead of completing their maturation journey.
Is there a clinical example of that?
The absolute classic is acute promyelocytic leukemia, or APL. It's caused by a very specific chromosomal translocation. This genetic mix-up traps the myeloid progenitor cells, preventing them from becoming mature granulocytes, which are non-dividing immune cells. They just pile up as immature dividing blasts.
But didn't APL also provide a really important therapeutic insight?
It did. It was a fantastic proof of concept. It turns out you can treat APL cells with a derivative of vitamin A, all-trans-retinoic acid, or ATRA. This drug actually forces the leukemic cells to overcome that differentiation block. They differentiate into mature, non-proliferating granulocytes. It was revolutionary because it showed you could sometimes treat cancer not by killing the cells, but by forcing them to mature and stop dividing.
Wow. Okay, and the third type of plasticity was transdifferentiation. This sounds even more radical, like cells completely changing their identity.
It is pretty radical. It's where a cell switches from one developmental lineage to a completely different one. It starts acquiring traits that weren't part of its original destiny, so to speak. Imagine a skin cell starting to behave like a nerve cell, or vice versa, although the actual examples are often more subtle switches between related epithelial or mesenchymal states.
So the cancer cell is almost sampling different identities to find one that helps it survive or spread better.
That's the emerging view. This ability to change phenotype to be plastic might be a distinct capability that cancer acquires, maybe separate from the initial driver mutations. It means that even if you target the original mutation with a drug, the cell might just transdifferentiate into a state that's resistant to that drug. It adds a whole new layer of complexity to treatment.
Definitely. Okay, shifting gears a bit, but still moving beyond simple DNA sequence changes. There's this whole area of non-mutational epigenetic reprogramming.
Yeah.
We're talking about chemical tags on the DNA, not changes to the A's, T's, C's, and G's themselves, right?
Exactly. This is huge. We now understand that critical gene activity, whether a gene is on or off, isn't just controlled by the DNA sequence. It's also heavily governed by these epigenetic mechanisms, chemical modifications to the DNA itself, or to the histone proteins that package the DNA. These act like switches or dimmer controls for genes.
And the main epigenetic mechanism implicated in cancer is DNA methylation. Can you walk us through that? How does adding a simple methyl group silence a gene?
So DNA methylation typically happens at specific spots in the DNA sequence, usually where a cytosine base C is followed by a guanine base. We call these CpG dinucleotides. Enzymes called DNA methyltransferases can attach a methyl group, CH3, to that cytosine. Now, often the promoter regions of genes, the bits that act like the on switch, are rich in these CpG sites. In cancer, you often see what's called aberrant hypermethylation. That means these promoter regions, particularly those of critical tumor suppressor genes that should be active, get excessively methylated. These methyl groups act almost like physical roadblocks, preventing the transcription machinery, the proteins that read the gene from binding. The result: the gene is effectively silenced, inactivated, even though its DNA sequence is perfectly normal.
So you can lose a crucial tumor suppressor function without any mutation at all. What are some key genes silenced this way?
Oh, there are many well-known examples. E-cadherin is a big one. That's the protein crucial for holding epithelial cells together. Silencing it helps cells detach and metastasize. BRCA1, the famous breast cancer susceptibility gene involved in DNA repair, is another common target for silencing by methylation.
Now, the really exciting part here the sources mentioned is that unlike a permanent mutation, this epigenetic silencing might be reversible.
That was the huge aha moment therapeutically. Because it's a chemical modification, not a change in the underlying code, it's potentially reversible. There are drugs called demethylating agents, like decitabine, that can inhibit those DNA methyltransferases or help remove the methyl groups. The idea is to chemically flip the switch back to on, reactivating those silenced tumor suppressor genes. It opens up a whole different avenue for treatment compared to trying to fix mutated DNA.
That's incredibly hopeful. And you mentioned the environment plays a role here too, like hypoxia.
Yes, the tumor microenvironment itself can actively drive epigenetic changes. Hypoxia, that lack of oxygen that develops as tumors outgrow their blood supply, is a major trigger. Low oxygen levels can actually reduce the activity of enzymes called TET demethylases, which normally remove methyl groups. So hypoxia leads to less demethylation, resulting in a general increase in hypermethylation across the genome, often silencing more protective genes. The tumor's own stressful internal environment actively reshapes its epigenetic landscape to its advantage.
The environment collaborating with the cancer. That leads us perfectly into the final new dimension mentioned, the polymorphic microbiome as an enabling characteristic. We're talking bacteria and fungi living in us actually influencing cancer. That sounds like sci-fi.
It might sound like it, but the evidence is becoming really compelling. It's a rapidly exploding field. The idea is that these vast ecosystems of microbes living in and on us, particularly the gut microbiome, aren't just passive bystanders. They can have profound impact on whether cancer develops, how it progresses, and, really importantly, how patients respond to treatments like chemotherapy and immunotherapy.
How does the gut flora actually do that? What are the mechanisms?
It's complex and diverse. We're seeing from association studies and even experiments using fecal transplants in animal models that there seem to be both good microbiomes that are protective and bad ones that promote tumors. Mechanistically, some gut bacteria might produce metabolites that mimic growth factors, binding to receptors on gut epithelial cells and constantly stimulating proliferation, directly feeding into that hallmark we discussed. Conversely, other good bacteria produce things like butyrate, a short-chain fatty acid. Butyrate can actually induce senescence or cell death in colon cancer cells, acting almost like a natural tumor suppressor produced by our own flora. So the balance of microbes really matters.
But it's not just local effects in the gut, right? The microbiome can influence things systemically far away from the intestines.
Absolutely. This is where it gets incredibly intricate. Take liver cancer, specifically cholangiocarcinoma. Studies suggest that gut dysbiosis and unhealthy imbalance in gut microbes can lead to a leaky gut barrier. This allows bacteria or their products like lipopolysaccharide, LPS, to seep into the portal vein, which drains directly from the gut to the liver.
Okay, so liver gets exposed.
Right. And once in the liver, these bacterial products can trigger inflammatory signaling pathways. This can lead to the recruitment of specific types of immune cells called granulocytic myeloid-derived suppressor cells, or GMDSCs. These GMDSCs are notorious for suppressing the activity of T cells and natural killer cells, the very immune cells we need to fight cancer. So an unhealthy gut microbiome can effectively create an immune-suppressive environment in the liver, helping liver tumors evade immune attack. It's a systemic effect orchestrated by gut microbes.
That's astonishing. And then there's the really cutting-edge idea of intratumoral microbiota microbes living inside the tumors themselves.
Yes, this is a relatively recent and quite startling discovery enabled by better sequencing techniques. We now know that many solid tumors, lung, pancreatic, breast cancers, for example, aren't sterile. They contain their own distinct communities of bacteria and sometimes fungi. And what's really striking is where they're found often localized inside the cancer cells themselves or inside immune cells within the tumor.
Wow. Are they just passengers or are they doing something?
The evidence is mounting that they're active participants. Functional studies suggest these intratumoral microbes can promote inflammation, help the tumor cells resist chemotherapy, and further modulate the local immune response. The big questions now are, where do they come from? Are they migrants from the gut or elsewhere? Or are they long-term residents? And crucially, can we target these intratumoral microbes therapeutically? It's a whole new frontier.
Okay, so we've got a rogue cell that's achieved immortality, it's become plastic, it's manipulating its epigenetics, and maybe even co-opting microbes. But like you said earlier, what ultimately makes cancer deadly for most people is its ability to grow really big and, more importantly, to spread.
That's right, growth and spread. These are the processes that turn a localized problem into a systemic, life-threatening one. It hinges on two key abilities: securing a dedicated supply line and executing the escape plan.
Let's start with the supply line. That brings us to the angiogenic switch. Why is growing new blood vessels angiogenesis so critical for a solid tumor?
It comes down to basic physics and physiology. A tiny clump of cells can get oxygen and nutrients just by diffusion from nearby existing blood vessels. But that only works over very short distances, maybe 100 or 200 micrometers, about the size of a pinhead or a small pea. Once a tumor gets bigger than that, the cells in the center start to starve for oxygen and nutrients. They become hypoxic and necrotic.
So a tumor hits a growth ceiling.
Exactly. It cannot grow beyond that small size unless it can trigger the formation of its own new blood supply. It has to flip the angiogenic switch to on, inducing neovascularization.
How does it convince the body to build blood vessels just for it?
Hypoxia is often the key trigger. When those cells in the tumor core become starved of oxygen, it stabilizes a protein called hypoxia-inducible factor 1 alpha, or HIF1.
Okay.
HIF1 is a powerful transcription factor. It gets activated and switches on the production of various pro-angiogenic factors. The most famous and important one is vascular endothelial growth factor, or VEGF.
VEGF is the signal flare.
It is. VEGF diffuses out from the hypoxic tumor cells into the surrounding tissue and finds its way to nearby existing capillaries. It binds to receptors, mainly VEGFR2, on the surface of the endothelial cells that line those capillaries.
And that binding wakes up the endothelial cells.
It activates them. These activated endothelial cells then start to produce and secrete enzymes, primarily matrix metalloproteinases, MMPs.
Ah, MMPs again, the molecular scissors that chew through tissue.
Precisely. The MMPs degrade the basement membrane, the sort of structural sheet surrounding the capillary, and the extracellular matrix, the scaffolding between cells. This allows the activated endothelial cells to break free, migrate towards the source of the VEGF signal, the tumor, and start dividing. They then organize themselves into new, albeit often chaotic and leaky, capillary sprouts that grow towards and into the tumor, bringing them much-needed oxygen and nutrients.
It's like the tumor orders its own plumbing. Is P53 involved here too?
It can be. Interestingly, functional P53 normally inhibits angiogenesis, partly by promoting the production of an anti-angiogenic factor called thrombospondin 1. So when cancer cells lose P53 function, which they often do, they also lose this natural brake on blood vessel growth, making it easier to flip the angiogenic switch.
Now, targeting angiogenesis, blocking VEGF, for instance, seemed like a really promising therapeutic strategy, drugs like bevacizumab. But the results have been, well, maybe not the home run people hoped for. Why is that?
It's a complex picture. The initial appeal was strong because you're targeting the genetically stable endothelial cells, not the rapidly mutating cancer cells, so maybe resistance would be less of a problem. And these drugs do provide benefit, often extending survival.
But not usually a cure.
Right. The problem is cancer cells are incredibly adaptive. When you block the main VEGF pathway, tumors often find workarounds. They might start overproducing alternative pro-angiogenic factors like FGF or PDGF, or they might switch tactics entirely and start co-opting existing host blood vessels instead of building new ones, a process called vessel co-option. They essentially develop resistance mechanisms that bypass the drug's effect. So anti-angiogenic therapy often slows the tumor down, but rarely eradicates it completely, especially once it's established.
Okay, so the tumor gets its supply line. Then comes the really dangerous part, the escape plan, metastasis. You said this counts for the vast majority, like 90% of cancer deaths.
It absolutely does. Metastasis is the ultimate cause of mortality in most cancer patients.
Yeah.
It's an incredibly complex and inefficient process, actually, but when it succeeds, the consequences are devastating.
It's described as a cascade. What are the main steps, starting with getting into the circulation?
The first major step is intravasation, invading through the wall of a nearby blood vessel or lymphatic vessel to enter the circulation. And interestingly, cancer cells aren't naturally very good at this compared to, say, immune cells. Leukocytes can slip through vessel walls in seconds using a process called diapedesis. Cancer cells often have to rely more on brute force, secreting those MMPs to degrade the barriers, physically pushing their way through. It can take hours, even up to a day.
And once they're in the bloodstream, it's not exactly a safe haven for them, is it?
Not at all. Step 2 is transport through the circulation, and it's incredibly perilous for a tumor cell. They face immense shear stress from the blood flow, anoikis, which is cell death caused by detachment and attacks from immune cells like natural killer cells. The vast majority of circulating tumor cells actually die during this phase.
So only the toughest survive.
Yes. Or perhaps the luckiest. Or those that travel in clumps, sometimes shielded by platelets forming little microthrombi. These survivors eventually get physically trapped, or lodge, in the first capillary bed they encounter where the vessel narrows. This physical trapping explains why metastasis often occurs in organs with dense capillary networks that filter the blood, typically the liver and lungs first, depending on where the primary tumor is, and also commonly bone and brain.
Okay, they've survived the journey. They're stuck in a capillary in a new organ. What next?
They have to get out again. Step 3 is extravasation, which is basically the reverse of intravasation, pushing or squeezing their way out of the capillary and into the tissue of the new organ.
And then the final and maybe hardest step.
Step 4, colonization. This is often considered the most challenging and rate-limiting step in the whole cascade. The cancer cell, the seed, has landed in a new environment, the soil of the target organ. This new microenvironment might be completely different metabolically, structurally, immunologically. The cell has to adapt, survive, start dividing again, and establish a new tumor mass, a micrometastasis that can then grow into a full-blown secondary tumor. Many disseminated cancer cells fail at this stage. They might die off or enter a state of dormancy, sometimes for years, before potentially reawakening later.
It's an incredible obstacle course. What are the key molecular changes that enable cells to even attempt this invasion and migration?
2 things are paramount. First, loss of cell adhesion. In epithelial cancers, which are the most common type, the single most critical protein is E-cadherin. It's like the molecular glue holding epithelial cells together in sheets. It also transmits signals that normally restrain growth. When cancer cells lose E-cadherin function either through mutation or, very commonly, through that epigenetic silencing by methylation we talked about, the cells become less tightly anchored. They can detach from the primary tumor mass and gain the motility needed to invade.
They unstick themselves.
Exactly. And second, they need to be able to chew through the surrounding tissue matrix. This involves ramping up the production and secretion of those extracellular proteases, especially the matrix metalloproteinases, MMPs. These enzymes degrade the components of the extracellular matrix and basement membranes, literally clearing a path for the cancer cell to migrate and invade through tissue barriers and into blood vessels. Loss of adhesion plus enzymatic degradation, those are key enablers of the metastatic journey. So I think this whole deep dive really underscores the sheer complexity we're dealing with. Cancer isn't just one broken part, it's a whole system gone wrong, constantly adapting and evolving. And acknowledging that complexity is forcing a major shift in how we try to treat it. We're moving away, thankfully, from the sort of sledgehammer approaches of the past towards much more targeted, sophisticated strategies.
Yeah, let's contrast those. The traditional approach, cytotoxic chemotherapy drugs like alkylating agents or antimetabolites, they basically just went after any cell that was dividing quickly.
Exactly. The principle was relatively simple. Cancer cells divide rapidly, so hit all rapidly dividing cells. But the problem, of course, is that many of our normal, healthy tissues also rely on rapid cell division. Think about hair follicles, the lining of the gut, the bone marrow-producing blood cells.
Which is why chemo caused all those awful side effects, hair loss, nausea, low blood counts.
Precisely. That lack of specificity meant significant collateral damage to healthy tissues. These drugs were, and sometimes still are, necessary, but they're blunt instruments.
So the newer approaches, the targeted therapies, they try to zero in on the specific molecular flaws that are unique to the cancer cell, the ones we've been discussing.
That's the goal. Instead of carpet bombing, it's about precision strikes. One major class is the tyrosine kinase inhibitors, or TKIs. These are typically small molecules chemically designed to fit into the active site of specific tyrosine kinase enzymes inside the cell enzymes like BCR-ABL or EGFR that are often hyperactive in cancer, constantly sending growth signals. By blocking that enzyme, you shut down that specific pathway.
And the other big category is monoclonal antibodies, MABs. These sound like engineered antibodies.
They are. MABs are laboratory-produced antibodies designed to recognize and bind to very specific targets, usually proteins on the surface of cancer cells or proteins involved in their growth signaling. They offer incredible specificity.
And they can fight cancer in different ways, can't they? Not just blocking things.
Right. They're very versatile. Some MABs work by directly blocking growth factor receptors. For example, cetuximab targets the epidermal growth factor receptor, EGFR, preventing the natural growth factor from binding and stimulating the cell. Others are designed to flag the cancer cell for destruction by the immune system. They bind to the tumor cell, and the tail end of the antibody then attracts immune cells, like natural killer cells, to come and kill the flag cell, a process called antibody-dependent cellular cytotoxicity, or ADCC.
And haven't there been advances to make these antibodies seem more human to our immune system?
Yes, that was a crucial step. Early MABs were often made using mouse proteins, which could trigger allergic reactions in patients. Technologies like the xenomouse mice, genetically engineered to produce fully human antibodies, have allowed the development of MABs that are much less likely to be rejected by the patient's immune system, making them safer and more effective.
Alongside these targeted drugs, there's been a revolution in harnessing the patient's own immune system, immunotherapy. Let's talk about vaccines first.
The basic idea of a cancer vaccine is to train the patient's immune system to recognize and attack tumor cells. We wanted to see tumor-specific antigens molecules that are unique to the cancer or highly overexpressed as foreign invaders. The most successful examples so far are actually preventative vaccines.
Like the HPV vaccine.
Exactly. Vaccines like Gardasil target the human papillomavirus itself. By generating protective antibodies against HPV, they prevent the initial infection that can lead to cervical and other cancers driven by those viral oncoproteins like E7 and E6. It stops the cancer before it even starts by targeting the causative agent.
But there are also efforts to make vaccines against existing cancers, using the body's own immune cells, like dendritic cells.
Yes, that's a major area of therapeutic vaccine research. Dendritic cells are the professional antigen-presenting cells of the immune system. They're incredibly good at gobbling up material, like tumor antigens, hopefully, processing it, and then displaying fragments of those antigens on their surface to T cells. The idea is to take a patient's dendritic cells out, load them in the lab with tumor-specific antigens, and then reinfuse them. These loaded dendritic cells should then activate the patient's T cells to recognize and kill tumor cells expressing those antigens anywhere in the body. It's about kickstarting a targeted cell-mediated immune response.
That sounds complex.
Yeah.
But perhaps the biggest game changer in immunotherapy recently has been something else. Immune checkpoint blockade. This isn't about showing the immune system the target. It's about releasing the brakes.
That's the perfect analogy. It was a truly paradigm-shifting discovery. It turns out that our immune system, particularly T cells, has natural brakes or checkpoints built in to prevent it from attacking our own healthy tissues too aggressively. Molecules like PD-1, programmed death-1, on T cells, act as such a brake when they bind to their partner, PD-L1, which can be expressed on other cells.
And cancer cells exploit the system.
They do, brilliantly. Many cancer cells learn to overexpress PD-L1 on their surface. When an activated T cell approaches, the cancer cell effectively shows that the PD-L1 stop sign. This binding engages the PD-1 brake on the T cell, shutting it down, paralyzing the immune attack right at the tumor site. The cancer creates its own little zone of immune privilege.
So checkpoint inhibitors just block that stop signal.
Exactly. Drugs that are antibodies against PD-1 or PD-L1 physically block that interaction. They prevent the cancer cell from hitting the T cell's brakes. This unleashes the T cells that were already present and primed to attack the tumor, but were being held back. The results have been remarkable, leading to durable remissions in some patients across a wide range of cancers: melanoma, lung cancer, kidney cancer, and more. It showed we could effectively treat cancer not just by targeting the tumor cell itself, but by modulating the immune system's radiation.
Incredible. Okay, let's tie this all together with the concept of translational research, getting these discoveries from the lab bench to the patient's bedside. The NTRAC model, as it's sometimes called.
Yes, bridging that gap is everything. And there's probably no better poster child for successful translational oncology than the story of chronic myeloid leukemia, CML, and the drug imatinib, perhaps better known by its brand name Gleevec. It really set the standard.
It started with a very specific genetic discovery, didn't it? Way back.
It did. Back in the 1960s, researchers in Philadelphia identified an abnormally small chromosome 22 in patients with CML. It became known as the Philadelphia chromosome. Later, it was figured out that this wasn't just a deletion, it was a translocation. A piece of chromosome 9 had swapped places with a piece of chromosome 22.
And this swap created something new.
It created a fusion gene. The break on chromosome 9 happened within the ABL gene, a tyrosine kinase, and the break on chromosome 22 was within the BCR gene. When they fused together, they created a novel hybrid gene, BCR-ABL.
And the protein made from this fusion gene was the culprit.
Precisely. The BCR-ABL fusion protein is a hyperactive constitutively on tyrosine kinase. It constantly sends proliferation and survival signals, driving the uncontrolled production of white blood cells characteristic of CML. The central insight, the translational leap, was realizing that if this single rogue enzyme was driving the whole disease, then maybe a drug that specifically blocked just that enzyme could treat CML effectively and with fewer side effects.
And imatinib was designed specifically to do that, to fit into BCR-ABL.
It was the result of rational drug design. Scientists screened thousands of compounds to find one that would fit perfectly into the ATP binding pocket of the BCR-ABL kinase domain, blocking its activity, but ideally not affecting other essential kinases too much. Imatinib was that molecule.
And the clinical results were?
Nothing short of revolutionary. Patients who previously had a very poor prognosis suddenly experienced dramatic and sustained remissions. It transformed CML from a fatal disease into a manageable chronic condition for most patients. It was the absolute proof of principle for targeted therapy based on understanding the specific molecular driver of a cancer.
But even with a success story that big, science never stops. Imatinib wasn't the end of the CML story, was it? New challenges emerged.
Absolutely. That's the nature of cancer biology and translational research. Every success generates new questions. Within a few years, doctors started seeing patients relapse because their leukemia had become resistant to imatinib. Researchers quickly figured out the main reason. The cancer cells had developed new point mutations within the BCR-ABL gene itself.
Changing the target so the drug no longer fits.
Exactly. The most notorious was the T315I mutation, often called the gatekeeper mutation. This single amino acid change in the kinase domain physically blocked imatinib from binding, rendering the drug ineffective. This spurred the development of second and third generation TKIs designed specifically to overcome T315I and other resistance mutations. Furthermore, longer-term follow-up raised other questions, like potential off-target effects. Some studies suggested a possible link between imatinib and cardiac dysfunction, perhaps because the drug wasn't perfectly specific and might inhibit other related kinases important for heart muscle function. It just highlights that even our best targeted therapies need continuous refinement and monitoring. The biological arms race continues. #outro.
Wow, what a journey. We've gone from the very foundations, a single mutated cell, the loss of gatekeepers like PRB and guardians like P53, through these incredible, almost sci-fi concepts of phenotypic plasticity, epigenetic reprogramming, the microbiome's influence, all the way to the sophistication of drugs like imatinib and the power of unleashing the immune system with checkpoint inhibitors.
It really covers a huge amount of ground, and I think the crucial takeaway for you, the listener, trying to grasp this evolving field, is that we've had to move away from oversimplification. Cancer isn't just one broken gene, it's a complex, dynamic system. The cutting edge of research now is really about trying to define what some call the complete integrated circuit of the cancer cell, understanding how all these pathways, mutations, epigenetic changes, and environmental influences interact. Only by understanding that whole circuit can we truly predict why a certain drug might work for one person but fail for another, or how the cancer is likely to evolve resistance.
That sounds like an almost overwhelming amount of information to gather and process for every single patient, though.
And that, I think, is the major challenge looming for the future. We're getting incredibly good at identifying all the genetic mutations, the epigenetic marks, the molecular profile of an individual tumor, using technologies like next-generation sequencing and microarrays that can assess thousands of things simultaneously. The bottleneck soon won't be finding the abnormalities. The real challenge will be developing and, crucially, affording and deploying truly bespoke, individualized treatment regimens. Imagine needing a unique cocktail of targeted drugs and immunotherapies, maybe even microbiome modulators, tailored precisely to the molecular fingerprint of your specific tumor at that specific time. Integrating all that data and translating it into an actionable, affordable treatment plan for every patient. That presents a monumental logistical, ethical, and economic hurdle that healthcare systems worldwide really need to start grappling with right now.